Hello students. Hello students. I am Dr. Vinod Shinde. In previous two videos, we have discussed about the physical and chemical agents that are used for control of growth of microorganism or control of microorganisms, in which we have discussed about the terms that are used in the as a chemical disinfectants or sterilization and other terms used for chemical disinfection purpose. Now, and in the last lecture, we have discussed about the characteristic features of the ideal disinfectants. Now we'll discuss about the actual chemical disinfectants that are in use. Of that, first one is the phenol and phenolic compounds that are used as a chemical disinfectant. Phenol or carbolic acid is not used nowadays in the germicidal action, but it still finds some use for that purpose, that is germicidals. The phenol and cresols are efficient germicides in fairly concentrated solution. Phenol is soluble in water, but most of the other members of this group are not only slightly soluble. Phenol or carbolic acid is firstly used by Joseph Lister in 1880s for aseptic surgery. Joseph Lister, as a surgeon, used this chemical for reduction for reducing the infection of surgical incisions and surgical wounds. He instituted the practice of applying a solution of phenol to surgical incisions and surgical wounds, and reduction of infection was striking. He observed that the phenol or phenolic solutions were suppressing the growth of microorganism or infections in the wounds, and the wounds are uh, recovering quite dramatically fast. Later, he developed the practice of spraying phenol into the operating room area to control infection. But phenol has an additional distinction of being standard against which other disinfectants of similar chemical structure are compared to determine their antimicrobial activity. Phenol can be used as a standard for uh, determining the disinfectant capacity of an unknown chemical, that is called phenol coefficient technique. Phenol and phenolic compounds are very effective disinfectants, and a 5% aqueous solution of phenol in water is is used to radially kill vegetative cells of microorganisms. Spores are much more resistant to this aqueous solution. Many derivatives of phenol have been prepared and evaluated for their antimicrobial activity. Antimicrobial activity is enhanced by addition of chemical substitution in a phenol ring structure. Glycyl Glycyrrhesorcinol is a derivative of phenol and is marketed in the solution of glycerine and water. It is a strong surface tension reductant, which may account in a part for its high bactericidal activity. Hexyl resorcinol preparation are employed as a general antiseptic nowadays. Phenol and phenolic compounds are mostly used in the disinfection purpose, but phenol, being corrosive in nature and cancerous in nature, It is avoided nowadays as it can cause skin cancer with a prolonged effective use of phenol. In the practical application, phenolic substances may be either bactericidal or bacteristatic depending upon the concentration used of phenol. Bacterial spores and viruses are more resistant than are in the vegetative cell form of bacteria. Some phenolic compounds are highly fungicidal or fungicidal. The antimicrobial activity of phenolic compounds is reduced at an alkali pH and by organic material. Low temperature and the presence of soap also reduces antimicrobial activity of phenol. Pure crystalline phenol is colorless. Solution of pure phenol have limited application. However, derivative of phenol diluted in detergent or some other carrier find use in many commercial antiseptics and disinfectant preparations. Combination of compounds of this class with a detergent result in the products with good disinfectant as well as detergent properties. Crystalline phenol or have a good antimicrobial activity. It is colorless, but a solution form of phenol is having a lower antimicrobial activity and limited application. The derivatives of phenol, some derivatives of phenol, phenolic compounds, when used with a detergent or some other compounds, have a application in commercial antiseptics and disinfectant preparations. In alkali pH and in the presence of heavy organic material, the phenolic compounds have lower antimicrobial activity, and at low temperature and in the presence of soap, the activity of phenolic compounds as antimicrobial agent reduces drastically. This can be recovered by using derivatives of phenol with the soap, which is having a good detergent and disinfectant capacity or activities. So these combination of phenols are used nowadays. Mode of action of phenol is that exposure of microbial cell to phenolic compound produces a variety of effects depending upon the concentration of phenolic compounds to which the microbial cells are exposed. It results in the disruption of cell, precipitation of cell protein, inactivation of enzyme and leakage of amino acid from the cell. Although the specific mode of action is not clear, a lethal effect of phenol is associated with a physical damage to the cell. The phenolic compounds have mode of action in different ways as per the concentration of phenol being used. In the low concentration to high concentration, it may result in the disruption of cell, precipitation of cell protein, inactivation of enzyme and leakage of amino acids from the cell. Though the antimicrobial or mode of action of phenolic compound is not clear, the phenols have physical damage to the cell and it is associated with the lethal effect of phenol on the cell. In the next lecture, we will discuss about the second chemical disinfectant, that is alcohol. Till then, goodbye and